first things first, obviously, does anybody have any L-serine on them? That would be very useful at the moment. So I was originally expecting to talk for 10 minutes, and then 5 days ago I was expecting to talk for 20. And when I checked the timetable yesterday morning to find out when I needed to be here, I've got half an hour all of a sudden. But now I'm actually on at the time when I only had 10 minutes. But I mean, I've tried to. I mean, I might talk for half an hour. I've not practiced; it's not my style. So we'll see. We'll see how we go. Okay. Well, anyway. So I guess this is where we start. Thank you very much for inviting us to your party. This invitation is kind of back to front, really. So what happened was I bumped into Gillian and Sandra in Boston at the GRIN meeting, and I think you're about to announce this conference, so you're really excited, and you told me about it, and I said, oh, Barcelona, I love Barcelona, I've got friends, and yeah, I could come, but this is kind of upside down here, because the RSVP kind of came at the beginning, because I got back in touch with you about 4 months later, and I said, oh, you know, you kind of invited me to Barcelona, and they were. Anyway, so I kind of sort of invited myself. I got stuck on at the end. That's fine. It's not really our receptors. No, that's cool. And it was really quick as well. So I had AMPA receptors are fast. I had a short talk. It was all good. But anyway, sorry, that was far too much of a digression. Let's actually get started. So I guess what I really want to talk to you about is this won't be news to the researchers and the clinicians, but the GRIN receptors are part of a wider family, including other receptors, GRIA, GRIC, and GRID. So this is just to kind of put you in a slightly wider context. I'm specifically going to be talking mostly about AMPA receptors, which is what we work on. So there are 4 genes, GRIA 1 to 4, and they're vital for normal signaling in the brain. And I'm going to talk a bit about GRIA disorder and the mechanisms of GRIA disorder in terms of variants, in terms of the experiments that we do. And ultimately, I've talked some work we've already done in terms of identifying precision therapies for GRIA disorder, and talk about a few ideas about which we might be moving on to shortly. Okay, so we kind of saw this scaling of our work in a couple of different ways yesterday, but this is my idea. So here's the brain. So if you imagine we're going to Google Maps it with our finger and forefinger and kind of zoom in. And if you start zooming in, you start to see that the brain is made up of connections of neurons all meeting up together. And you zoom in further and you find the connections between these neurons are made up of synapses, where a presynaptic release of neurotransmitter activates receptors on the postsynaptic side. And then zoom in further, you start to see these proteins appear. And we know we have a very, very good understanding now of what these proteins look like. And ultimately, we can know exactly where the specific parts of the specific amino acids that make up the protein, where they sit, and have a pretty good guess about what they do. So we have a really, really good molecular understanding. And this is a zooming in factor of probably about 100 million or something. So we really, really are tiny, tiny, tiny. Okay. So the NMDA receptors, the GRIN genes, are part of a receptor superfamily, and the GRIN genes are here. So they're GLUN1 is slightly less related to GLUN2 and GLUN3 than they are to each other, and but there's also other gene families as well, and this includes the GRIA genes, which are AMPA receptors, GLUA 1 to 4, and also the GRIC genes, GLUK 1 to 5, and the slightly funky delta receptors. And they've all got similar genetic sequences. That's why we position them in a tree like this. But they've also got pretty similar three-dimensional structures and also mechanisms of gating. So you see that all the receptors sit in the cell membrane here. So they've got these bits of protein that effectively punch a hole in the membrane and that allow them to control the entry of ions inside the cell. And they also have, most importantly, just outside the membrane, a ligand binding domain that binds glutamate mostly, but for the GLUN1 subunit, it binds glycine or D-serine, and also for these delta receptors, also bind glycine and D-serine. So they're all glutamate receptors, but there's a little bit of wiggle room in their ligands. And the overall structure of AMPA receptors and kainate receptors and delta receptors has a subtle difference in the most distal part of the protein, and this probably helps them reach across the synapse and actually hold on to the presynapse. Whereas the NMDA receptors are slightly more contained. And that is why the amino terminal domain of the NMDA receptors is more important to receptor function than the amino terminal domain of these other members. So there are similarities, but also a few, even at this scale, a few differences. So, I mean, you'll know this, NMDA receptors come in a few flavors. So they're what we call obligate heteromers, which is a fancy way of saying you can't make a receptor out of the same 4 of the same subunit. You need different subunits. And so they must contain 2 GLUN1 subunits, and they also contain 2 either GLUN2 or 3 subunits. And there's a bit of variety. They have several splice forms. So the GLUN1 subunit has got splicing in its amino terminal domain, which gives, my first slides made at 1 o'clock in the morning, little typo there. I'm sure there'll be others. Okay, so the amino terminal splice form gets called terms A and B, and there's also some splicing in the C tail as well, which gives forms 1, 2, 3, and 4. Okay, they're not modulated by auxiliary subunits, so, or at least none have been discovered. It's actually slightly harder to solubilize NMDA receptors than some of the other glutamate receptors. And so by the time you've solubilized it enough to get it out of a synapse, you've probably made any potential auxiliary subunits fall off. So there may be some that we haven't discovered, but in general, they're not critical for the way NMDA receptors work. And kind of looking at all the combinations that this can potentially produce, if you've had your child's variant assessed for its functionality, almost always, if it's 2A, it will be in this combination here, this diheteromer. So of GLUN1 with the lacking the splice form here, so form A, and of the form number 1 of the C tail. And so it's just one combination that's been looked at here. And the same for 2B and 2D. I believe that's almost certainly what's happened. Apologies if I've misrepresented anyone's research there. But there are other forms as well, of course, based on these splice forms. So you can have the 1B, you can have an extra chunk of protein incorporated here, or you can have triheteromers, so receptors with 2A and 2B. You can have the different C-tail forms, and you can have receptors with GLUN3 as well. So there are lots of different combinations, but not nearly so many combinations as we have with AMPA receptors. So these have 4 subunits, and they can be a mixture of multiple of the GLUA1 to 4 subunits. They can form homomers. You can get pure receptors just with one form, or typically they contain 2 GLUA2 subunits in the brain and a couple of other subunits. They also have alternative splicing, so different chunks of protein that can be switched in and out in terms of their structure. And they do have lots of auxiliary subunits. So they have families called the TARPs, cornichons, GSG1L, and CCAM. So a range of different auxiliary subunits that sit around, typically around the membrane regions of the receptor, and they kind of sit in here, and they are important for trafficking, they're important for modulating function, and they're basically absolutely crucial to the way that AMPA receptors work. And what this means is that the table for AMPA receptors is a lot bigger. So again, you can form homomers. So if you wanted to look at the functional effects of a variant in GLUA1 or GLUA2 or GLUA3, GLUA4, you can make a receptor purely out of those subunits. And that's really handy. When we come to assess the effect of variants, we don't have to worry about a phenotype being diluted by the presence of other subunits. It kind of makes our lives a lot easier. But of course, they have these auxiliary subunits, and all native AMPA receptors, well, as far as we know, all native AMPA receptors contain some kind of auxiliary subunit. And so when we express them, we can make homomers with a TARP. This was the original one discovered, TARP gamma 2, and again, homomers of GLUA2 with gamma 2. But the receptor is normally heteromers. So you can also have 1, 2, 1, 2 with these auxiliaries. And, you know, this is a common one in the hippocampus, probably, GLUA1, 2 heteromers with gamma 8 and cornichon 2. And then you can have CCAM44, you can have multiple TARPs, you can have different subunits, and et cetera, et cetera, et cetera. And there's probably, well, there will be definitely over thousands, literally thousands, once all of these combinations. And if you throw in the alternative splicing as well, there are thousands of different potential combinations of AMPA receptors. And at the moment, when your inbox seems to be having a fresh GRIA variant arrive every week, you can't do that. There's no, you know, it will slow you down so much trying to find things that are probably just going to behave a slightly different version of the same. And so we need to come up with a standard condition. And right now, what we've chosen to go with is homomeric receptors in the presence of a TARP. Okay, and then that we know that we do that for every condition. There may be reasons to go and explore certain other combinations if there's a particular reason to, but this is our baseline condition, and this is what we choose to look at. Okay. Right. So yes, so we're here. Well, I'm here because as well as GRIN disorder, there's also GRIA disorder. So it's the same general principle: de novo changes in a glutamate receptor gene that causes some kind of neurological issue. And in terms of identifying GRIA disorder, well, I guess the process is kind of the same as it is with GRIN. So you have a child who's unexpectedly unwell. There's no obvious reason as to why this should be, in which case it's a really good idea to check for de novo changes. And the reason why I think the discovery of GRIA disorder has kind of lagged a bit behind GRIN disorder is that we weren't on enough of these lists early enough. Okay, and so, for example, this is a company called GeneDx, American company that do a lot of these kind of studies. They have a panel of genes called the Epi-expanded panel, which has got about 1,700 genes on it. Okay, and I mean, which is remarkable because this is almost 10% of the genes in the entire genome. So there are an awful lot of ways of breaking the brain, and the reason I'm flagging this up and why those little ones there are in red is that you also find the glutamate receptor genes in amongst this huge list. And of course, you know about GRIN1, GRIN2A, GRIN2B, GRIN2D, but the AMPA receptors are also there as well, 2, 3, and 4, and GRIC2. And actually, really just the last couple of weeks, again, things in my inbox and meeting parents here as well. It seems that there's really GRIA1 should be on this list as well, because while there have been only really a handful of GRIA1 patients, they never really had seizures before. I think there's a couple of patients that just cropped up, one of which has definitely had seizures and one of which was possibly kind of seizure-like activity. So trying to document this and report it and trying to get the genes added to the list means that you look more and you find more. So that's something that we're kind of focused on at the moment. And it's also important to realize that at the moment, I mean, this particular, I put this slide together originally because we had a GRIA2 patient, and GRIA2 wasn't on any of the other epilepsy panels. They had other much simpler epilepsy panels with far fewer genes, and it was only present on this one. And if they hadn't gone back for a third time to get the final mega test done, they wouldn't have discovered what the problem was, and then we wouldn't ultimately have been able to help them, which is what I'm going to talk about in a bit. Okay, so GRIA disorder. So this is kind of defined as a genetic variation associated with neurological disease, and it's been identified in each of the GRIA genes. So around 200 cases of GRIA disorder have been identified. There will definitely be more than that because we just haven't been looking. And I can say this, that number may surprise you a little bit, but I know for a fact that there are a couple of different papers that I think are in press. And so within the next month or so, depressingly, I'm not on any of them, unfortunately, but some other people have had a really good time. So so there's a paper with 50 variants across all of the subunits and one specifically about GRIA3. And once those are out there, that's going to take the published cases up above 100. And in terms of other ones we know of that are not yet published, this is going to take the total number up to about 200. In general, it seems like this is kind of the way it's panning out, that there'll be less GRIA1s than there are GRIA4s, than there are GRIA2s, than there are GRIA3s. So that's generally the way it's working out. One of the reasons why GRIA3 is right at the top is because it's X-linked. And so males only have one copy. And so you can potentially have a healthy mother who has one compensatory copy, passing on just out the blue, having a child with neurological disease. In terms of where these numbers are ultimately going to end up, I don't believe that they will reach the numbers of GRIN2B, but for example, GRIA3 could certainly start approaching the levels of GRIN1 and GRIN2A, but it's just too early to say. In terms of where GRIN2D would sit in this list, I suspect GRIN2D would be around more this kind of level, but I mean, I haven't seen all the latest stats. That's just a kind of a feeling. And as well as the genes being related, the symptoms of the patients are also related. I mean, this is kind of very broad. I know we had an excellent talk on subdivisions of epilepsy and so on over the last couple of days. But the key symptom is intellectual disability. This is pretty much ubiquitous. Around half of patients have seizures. I mean, that can, that's a fairly blanket term as well. Things like abnormal muscle tone and movement. Again, these are hugely diverse fields, and I've just lumped them into one column on a table. So I'm not saying that they look exactly like each other, but you could characterize some of their symptoms under this umbrella. And there are autistic features as well. And a few other cases, for example, ADHD has been seen in a few cases of GRIA2, schizophrenia and GRIA3. I mean, this is kind of slightly aside. These patients are not within the cohort that we look at as a child, but there are some big screens of genes linked to schizophrenia found GRIN2B, and it also found GRIA3 within that group. So in terms of more, sorry, don't quiz me on this. This is purely from our clinical colleague, but what he's beginning to see is that startle, or like hyperexlexia, so overreaction to noises and so on, seems to be more common in GRIA disorder than GRIN. And there are certain seizure syndromes that are found in GRIN disorder that don't seem to turn up in GRIA. So there are divisions, but there's certainly a lot of overlap. And so what we do is we look at these variants as they come in, and we try and work out in exactly what way the receptor is defective. And we've heard a lot about these studies. I won't dwell on it, but typically with our collaborators, they do some xenopozoocytes, so putting the DNA into this enormous cell, getting loads and loads of channels, and being able to do relatively straightforward electrophysiology. What we do is we get it into HEX cells. These are cultured human cells, and then we just take a small little bit of the membrane, and that allows us to have really tight electrical control on these channels, which is, I'll go on to show you, is essential for AMPA receptors. You can also, of course, do viral transfection. You can start to try and knock this into tissue, but unfortunately, what we don't have is a decent transgenic mouse. Ideally, we would have a transgenic mouse that had a phenotype that we could monitor, and they're just not out there yet. So that's really important as we go forward, trying to work out which therapies we can develop. Okay, and this is kind of what the kind of experiments we do. So variants, they break AMPA receptors in different, there's no other way of saying it. AMPA receptors work perfectly well, and something has changed. So the reason why it's so important to do experiments like this is that as with GRIN disorder, there's a huge spectrum. You know, the most severe patient is in the pantheon of all neurological patients is absolutely miles away from the mildest one, and there really is not necessarily that much similarity between the individual cases. So in GRIA2 disorder, for example, you can have adults with high functioning autism, basically living independently pretty much, to some cases of death due to epilepsy in young infants. And so we need to work out what each individual variant is doing to try and understand why the particular symptoms of the patient are the way they are. And so these are de novo changes as well. So there's no inheritance. So basically, because the numbers are still so low, even though there are a few variants crop up repeatedly, that if you get diagnosed with a variant, it's likely you'll be the only person who's got that variant. So they all need to be checked out. And yeah, we're trying to marry up the symptoms of the patient with the effect on the receptor. So just a couple of examples here. These are kind of some record breakers. I'm a geek. And so we have variant 1 here that's somewhere in the ligand binding domain. And this has the fastest desensitization of any receptor I've ever seen, possibly any receptor at all, but certainly any glutamate receptor I've looked at. So here's the wild type response. This GLUA2 response has a time constant of around 10 milliseconds in the presence of a TARP. This loss of function variant has a time constant of about 100 microseconds. So it's open and it's gone just like that. And of course, this would obviously lead to deficits in excitability between these synapses, these junctions between neurons. Variant 2, on the other hand, which is present somewhere in the pore region here, has the least desensitization of any receptor I've ever seen, and I'll show you that in a sec. This is exactly the same record, actually. This black trace here is the same on both sides, apart from here, when glutamate's applied, the receptor just sticks open, and then a very, 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 very slowly unbinds glutamate. So this has an incredibly high affinity. And it's no surprise to find that this patient has had seizures, and this patient has more autistic type symptoms and so on. So there's already a kind of a division starting to appear. Okay, yeah. So I did my PhD in biophysics at Imperial College, and I went to UCL, and I've been looking at the electrophysiology and the pharmacology, the protein science of glutamate receptors for a long time. And yeah, I love it. I mean, I'm very lucky to do what I do. But when a variant comes into our inbox, we're of course very, very aware that there's a sick kid on the end of the line, and we acknowledge that, and we won't forget that, and we're inspired by it. But yeah, I mean, your kids have some really cool ion channels. I've just got to say that as a geek. So let me show. And these are literally, this is fresh data. These are variants that have arrived in the inbox within the last month, and we synthesize them when we've had a look at them. So I think we've kind of mentioned that it's possible if you have really, really tight electrical control of a patch, you can look at the behavior of an individual ion channel. So just literally one protein complex opening and closing. And this variant here has an open probability of like 1.00000. You know, this thing, it just stays open. When glutamate hits the channel, it opens every single time and it stays open for ages. And so if somebody tells you, oh, I had a patch with 3 channels in, you're like, how can you be sure? And it's just like, no, you really do, because there's 1, 2, 3, 1, 2, 3, 1, 2, 3. And even when this should have added noise from all of the openings at once, it's just literally just open the whole time. So I'm geeking out. And of course this patient is very sick, but having looked at this experiment, I was able to inform the clinician and the mother that yes, this is certainly gain of function, and I believe that she has now started. Well, I'll come on to precision therapies in a moment. Even cooler in a way is this one here, which so the scales are a little different, like the time scale is different because this one closes very quickly, but the vertical scale is the same. And so this is like Godzilla. This is like this channel will swallow these 3 for breakfast and have those for lunch and then have these for tea. It's just absolutely enormous. I never, if someone had told me, could you make an AMPA receptor that had 150 picosiemen conductance, I would tell them certainly not. And yeah, so this is even in the absence of auxiliary subunits. So there are fringe benefits for geeks in doing this kind of research, as well as knowing that you're helping people. So, okay. And so our work at the moment kind of sits like this. So we are collaborating with some clinical colleagues at the Institute of Neurology and in the University of Copenhagen. And at the moment, we are looking at over 40 GRIA2 and GRIA4 variants to try and work out exactly what might be wrong with them. And this is kind of where we stand at the moment. I should say I haven't done any statistics on this. The statistics come at the end, because if you start doing statistics in the middle, you can be tempted to kind of, oh, I'll have a few more of those, and it's not the done thing. So this is all qualitative assessment of the data. And certainly it seems that for this, like the receptor I showed, deactivation is slow, desensitization is reduced. Clearly that's a gain of function. And there are 12 GRIA2 variants and 4 GRIA4 variants that seem to have this phenotype. Desensitization accelerated, that other one I showed you, which is obviously a loss of function. The channel can open, but then it shuts down very quickly. There are 8 of those in GRIA2. Ones with no current at all or greatly reduced current, just in terms of magnitude. There are 4 of those for GRIA2. And this is kind of an interesting one. The deactivation is a bit slowed and desensitization is intact. In actual fact, probably what happens with these receptors, they have a very high affinity, but of a really slow, desensitization is intact despite their high affinity. And so they bind glutamate, they open once and they stay shut. And so this is an interesting kind of mixed type phenotype one. We need to do some more work on this, but I suspect actually it does work out a gain of function when you think about its role within a tetrameric receptor. But there are 6 different variants like this in GRIA2. So this is something that we're keen to look at. And then certain ones with, we have our one-off GRIA4 variant here, Godzilla, has an increased conductance. That's almost certainly gain of function. But there are others which don't seem to have a functional change, or it's too early to say. And of course, the as yet untested ones. I mean, how many GRIA patients actually are there on planet Earth? I'm willing to bet probably 100 times more than we know about right now, and more than 100 probably. So so there's a lot more work to do. Am I okay for time? I'm really sorry, I haven't practiced. I've got no idea how long I've been talking for. The one thing I said, get your phone out and look at it. But no, I'm afraid, so I'm guessing. Okay. Okay, so this was stuck in because I've already spoken to some people in the audience when I spoke about this before. So maybe I'll kind of go a bit quicker. But this is our first patient ever. So we have been looking at AMPA receptors for forever, and about 2.5 years ago, we got contacted in a roundabout way by GRIN, the American. I'm sure you know them, and they were having contact from a few GRIA patients, and they thought, well, maybe we could look at them too. And so they got in contact with us because they knew what we did, and asked if we'd be interested in potentially working with them. And yeah, absolutely. I like looking at proteins and stuff, but I got into this as I thought I'd do medical research, and nothing until the last 2.5 years that I've done has got anything to do with medicine whatsoever. So this has been good. Okay, so this patient, the first clinical signs were episodes of back arching and vomiting. 9 months, started really going downhill, showing decreased interaction and worsening back arching. Had regular seizures from 12 months of age, leading to treatment with levetiracetam and phenobarbital, but the seizures kind of kept on going. Global developmental delay became more obvious, and there were worsening GI symptoms, and he was designated as failure to thrive, and there's an example of one of his seizures there. And it was around about this time, once he was already a year old, that the diagnosis finally came back. So this was the patient. They wanted to work out what was going on. The screens were negative, and it was only when they made the really big screen that they actually finally found out what was actually going on. Now, as soon as they told us about the site of this variant, we instantly knew where it was. This is a pretty famous site within glutamate receptor fields. This is the most conserved part of the receptor. And certainly when there's a T in this site, we knew that the receptor was overactive. And but no, we hadn't really looked with a V before, so we wanted to check it out. So what was already known is that the patients have neurodevelopmental delay, intellectual disability, ASD, seizures. However, previous variants in the GRIA2 gene have been shown to be mostly loss of function. So we suspected it was gain of function, but the patients are normally loss of function. So we wanted to have actually get the protein in our systems to work out what was going on. And the advantage, of course, of working out if it is gain of function, there's a drug called perampanel that will bind to AMPA receptors and inhibit their currents. And because it's already an anti-epileptic and we had an epileptic patient, it was a really obvious thing to try. However, if this variant proved to be loss of function, it wouldn't be a recommended thing to do because you have inhibited AMPA receptors that you're making smaller. So we didn't want to do that. So we wanted to do some electrophysiological analysis. So the same kind of experiments I showed before. In this situation, not as severe as the one I already showed you, my favorite one. This is deactivation is very, very slow. So it binds glutamate and it takes 5 or 6 times longer to let the glutamate go. And desensitization is reduced as well. So if you jump into glutamate and you hold it there, the receptors have a safety mechanism in which they can turn themselves off, even though they've still got glutamate bound. And that was defective. The wild type receptor goes all the way down to pretty much nothing, whereas the variant is active still half the time, and glutamate potency was increased. So all of these properties came together to say it was gain of function. And so okay, let's see what perampanel does. And perampanel did inhibit A643V, but at a reduced potency. So perampanel on the wild type is down here with an IC50 nearly 100 nanomolar, whereas up in our variant it was over a micromolar. So it was kind of about 10-fold weaker, but nonetheless it did inhibit the currents. Okay, and probably one of the reasons why it's less effective against the variant is that it binds here. The variant position is right next to where perampanel binds. And this is a question that maybe came up a little bit earlier. If you have a variant that is causing a problem, it's likely to be in an important part of the channel. And that's where drugs are likely to bind as well, because it's an important part of the channel. So so probably that limited the efficacy of perampanel. But nonetheless, there's nothing we found that said it shouldn't be safe to start treatment. So we recommended they did. And the results were pretty good. Seizures were reduced to less than 3 per month. Even though he was quite a young child, he had a really high dose of perampanel. There was some kind of metabolic reason as to why that was deemed to be safe. And this allowed him to successfully wean levetiracetam, which the family hated, and they really wanted him off it. And so they were really, really pleased to be able to do that. And you can see that the frequency of the seizures before versus after is greatly changed. I should also obviously point out that the modified Atkins diet, so ketogenic light diet, was also instigated, and you can see that probably had at least part of the effect as well, because there was a bit of a drop off even before perampanel was started. Okay. So this was kind of to be expected, to be honest. I mean, this is the anti-seizure drug, and it directly acts to counteract the variant. It was kind of a really, really obvious thing. But what we were interested to see, and we're very keen to try and monitor, was were there any benefits beyond the seizure control? Because if you have seizures and you dampen down AMPA receptors, it can stop your seizures, but it dampens down everything, and maybe it's not that great for the rest of your brain. But for this patient, of course, all of his AMPA receptors were boosted, and so the dampening effect of the perampanel might stop his seizures, but also potentially help to bring the rest of his brain into a better balance. And that indeed seemed to be the case. So the GI symptoms were no longer of concern to the family. His GI meds were stopped. Failure to thrive resolved. So he was really, really tiny. at one point, but then kind of went up to a much more acceptable weight. Physical development accelerated, so he's able to sit up, so able to weight bearing quadruped, grasping for things, reaching for things, and was moving on to moving. I should say these details are probably about 18 months out of date. I think we're going to get an update quite soon, but so I'm not quite sure exactly what is going on right now. And also the interaction was increased as well. So facial recognition, eye contact, visual tracking, could understand instructions and could verbally respond to yes and no questions. So it was, the result, of course, was remarkable, but you couldn't call this a trial. This wasn't even an N=1. This was just a case study. You know, we had a reason to try a treatment, and the patient desperately needed help, and we just did it, and this is what happened. You know, so I'm not claiming that any patient that has a precision therapy will treat their main symptom and all of their other symptoms as well. But certainly in this case, it was just a great result. That's all I can say. And actually, one of the reasons I was worried about perampanel is that it has some quite nasty side effects in terms of mood modulation and so on. So, yeah, basically behavioral problems and even aggression. But actually, he seemed to be really, really cheerful after his perampanel dose. It was almost like he'd taken serine, I guess, something like that. And again, you know, this is we couldn't have asked for a happier, better result. But it's not. I wouldn't call. I mean, yes, it's science, but it's not clinical science. Like the experiment worked in the patient, but it's not. You know, there are limitations. Okay, so the good thing is actually the main title of my talk was pretty misleading. I never got around to quite as far. I wrote a title that my experiments couldn't match in time. So we're going to peter out anyway. So I think I've probably been talking quite a long time. But just in general, just thinking about the potential for precision therapies in GRIA disorder. So GRIN drugs in blue and the GRIA drugs in green. So in terms of gain of function, you can block the channel with ketamine, memantine, and amantadine. There are no competitive antagonists, ones that bind to block glutamate binding, but there are non-competitive antagonists, perampanel, which I talked about, and also ridiprodil as well. In terms of loss of function, we have L-serine, and that's kind of basically it, really. So far, so you know, we'll see. Someday you'll tell us. But okay. Anyway, and I guess so. The point I want to make, and the point I'm really glad that you invited GRIA, is that AMPA receptors and NMDA receptors work together in the brain. So the fundamental difference between them is that AMPA receptors bind glutamate quite weakly, which gives a really short signal, whereas NMDA receptors bind glutamate much more strongly, which gives a long signal. So it kind of looks something like this. AMPA receptors, they bind glutamate, but then they let it go really quickly. So if you have 5 quick signals coming into a neuron, the AMPA receptors can respond 5 times. They pass on that level of information. NMDA receptors lack that kind of precision, that kind of fidelity, but nonetheless, they obviously pass a much bigger signal. So it's all important. They all work together. NMDA receptors, the synapse, are normally blocked by magnesium and require coagonists like glycine or D-serine. So, and AMPA receptor activity can help relieve this magnesium block, so to help NMDA receptors get activated, and can also, the evidence is possibly less strong, but it can also produce D-serine release. And so AMPA receptor activity helps NMDA receptor activity. The other way around, and slightly longer term, NMDA receptor activity and the calcium that comes through NMDA receptors can modulate AMPA receptor numbers. So they have a very symbiotic relationship. And what this potentially means is, we talked about precision therapy, AMPA drugs on AMPA, NMDA receptor on NMDA. Is there such a thing as a semi-precision therapy? So given the close physiological relationship between AMPA receptors and NMDA receptors, could drugs that target one class of receptor be useful for disorders of the other? And I mean, they could be a thing. If they are, nobody's called them that before. So I'm gonna own that. So anyway, just to kind of point out what, you know, just to really spell out the point. So if you have GRIN gain of function, then your NMDA receptors will be more sensitive to AMPA receptors than healthy NMDA receptors. That's almost a given. So if you were to try perampanel in GRIN gain of function patients, it has perhaps a slightly better chance than any other random drug about being effective, because it's kind of acting on the same systems. Similarly with GRIN gain of function, GRIA gain of function. A drug like memantine, for example, the gain of function AMPA receptors will activate NMDA receptors inappropriately. So if you can inhibit the NMDA receptors, then maybe AMPA receptors would be they won't be fixed, but at least part of the deficit will potentially be fixed. And I mean, and this was kind of really where it came from. So we actually had an email from a GRIA mum who said, well, L-serine looks like it's doing something. I have a loss of function GRIA2 child. Is there any point trying serine? And I started writing an email back saying, thanks for your message. You know, while serine looks promising, it's not on the same receptors, they're related. And as I was going through, I thought, well, actually, you know what, they do work together. And maybe if you have GRIA loss of function, then L-serine is something that should be looked at, because your loss of function AMPA receptors result in NMDA receptor hyperfunction downstream. So if you can boost the NMDAs, then you can't fix GRIA disorder, but part of GRIA disorder can be fixed. And so this was kind of the hypothesis. And this is why the question mark on the end of my title is now a really big question mark, because I've not actually got around to doing any of this yet. Okay, so yeah, there are two answers. And it could be. We will see. But just to, I mean, and your talk, you're already doing lots of this stuff, I think, in terms of the simulations and so on. So yeah, so the future plan is to develop simulations to mimic the interplay between NMDA receptors and AMPA receptors. And the good thing about this is that you only need to worry about postsynaptic currents, membrane potential, and calcium influx. And deserine release, glycine, short-term potentiation, long-term potentiation, spike time dependent, plasticity, long-term depression, glutamate receptor composition, spliceosome dependence, back-propagating upstream potential. And so basically, this is one long list of an excuse as to why I haven't got anything to show you. Because when we started looking at it, I suddenly realized, my God, neurons are more complicated than hex cells. Why did nobody tell me this? And the actual setting up of these simulations is really critical. We've got to get it right, because the point is, once we built the model, it's actually going to be very simple to plug in the aberrant properties of the loss of function, gain of function, GRIA variants to see how these behaviors are modified by the variants, and then of course use these models to see what can we predict would happen if we tried a GRIA drug on a GRIN disorder or a GRIN drug on a GRIA disorder. And that's something that we are actively, I was in frantic conversations with a PhD student in the lab at the start of the weekend going, I've got nothing to show them, how's it going? And he sent a picture of me, a picture of himself in the pub with a big line of beers in front of him and said, no, it's not my problem. So anyway, that is where we've got so far, which is not too far, but I hope you now understand a little bit more about GRIA. And so just to summarize, yeah, GRIA and GRIN are related proteins, related disorders, lots of related symptoms. We're looking at a lot of de novo GRIA variants to try and work out exactly what's wrong with them. And we know for a fact, and you're showing as well with all your great research, that precision therapies are showing promise as in treatment of these disorders. And we're excited to explore the possibility that there could be an overlap in the treatment of both GRIA and GRIN. I really need to flag this up because this is really important. So for any clinicians who happen to come across a GRIA patient or any GRIA patients themselves, you should contact Alan Byatt. He's building a new registry at the moment. He's overseeing, I said there was not much science in our perampanel story. He's adding some science to that. So any gain of function variants we can identify for him. He's doing a much more structured study on the potential benefits of perampanel. So really keep this email address in mind because that is kind of the future of GRIA. And final acknowledgements, UCL are collaborators at Copenhagen, that's where Alan Byatt is, and the ION, and these doctors in Michigan who help with our patient. And thanks very much for having us. If I can say us in terms of GRIA, because I mean, we don't have our own foundation at the moment. So I would say if you were to speak to CureGRIN, they would say that having us involved with GRIN has been of benefit to them, certainly. Adds a bit of variety, and also lots of the researchers are the same as well. You know, you're going to start diluting your research power if there are multiple of these major centers. And so we promise we'll hoover, we'll clear up the broken glass and wipe over the table and so on. But if it would be potentially possible for GRIA to join in with GRIN Europe as well as CureGRIN, then I think that would be excellent. So, oh yes, and final, sorry, very, very final thing. We are, you should come around to ours. So we have a mini conference that's coming up. It's scheduled for March the 2nd in London. So if anyone wants an excuse to come and visit London, then yeah, kind of get in touch. It's only one day, so there probably won't be as much time to talk, loads and loads of pure science. But certainly in terms of getting families together and so on, then I'm really sorry. I'm worried I've talked far longer than anyone, having been given the shortest time. But anyway, thank you very much.